Next speaker, we have Aure le Gilles. He's a assistant professor at Princeton University's Department of Applied Mathematics. And Mark's research interests are generally in numerical linear algebra, applied statistics, and applications to Cryo-EM. It's
great. So thanks for the introduction and thanks for the invitation. So today I'm going to talk about joint work with Amit Singer, who is my former postdoc advisor, who's also at Princeton, on reconstructing conformational states and free energy landscapes from Cryo-EM datasets. So let me give a 30-second introduction to Cryo-EM for the uninitiated. So a Cryo-EM experiment goes like this. You have to purify a sample to make sure there's only a single type of biomolecule in there, dump that sample into a grid, freeze it, and stick it into an electron microscope. Okay, and what you get out of the electron microscope is a big image that looks like this, a tomographic image. And inside of it, there's millions of sub-images of the same structure, but viewed from random direction. Okay, so unlike the X-ray talk we just saw, we actually observe sort of the phase information. So we can look at the images in spatial domain, and they look like this, the one on the right. They are very extremely noisy images. And the basic Cryo-EM reconstruction problem is to take sort of millions of images that look like that and to reconstruct a single three-dimensional structure. And so, as I said, the most important feature of these Cryo-EM images is that they're extremely noisy. And because they're extremely noisy, we typically need hundreds of thousands or millions of them to do a single reconstruction, which gives rise to gigantic data sets that are in the order of hundreds of gigabytes or terabytes for a single reconstruction. So I'm going to talk about heterogeneity analysis in Cryo-EM, which is what we call the reconstruction of ensembles of conformation from a data set. Comes in two flavors. There's a discrete kind, which typically arises from when we're trying to image a protein complex, and we're observing it in different states of assembly. And then there's the continuous version, which is maybe the more interesting one, where we're observing really the same structure, but some motion of the structure. And so this is a very popular topic in the Cryo-EM literature these days. There's been really dozens of approaches and papers on it in the last few years. So some of them I highlighted, Cryo-Dragon, which in my mind is the real first one to use deep learning methods to solve this problem, and it has become a very popular topic now in the deep learning literature. So here's a mathematical description of heterogeneity analysis in Cryo-EM. So what we're trying to recover is really these states, denoted by x's on the left. And really what we're imaging is the Coulomb potential here. And I'll think about this state x as being a three-dimensional function that I discretized on a voxel grid. And so the x's are related to the observable, which are the images as follows. So each of them is rotated, shifted, and projected into 2 dimensions. So one particularity, as was mentioned earlier, is that in Cryo-EM, we typically do not know how this particular state was rotated to in this image. So something we need to infer. After this projection, the image is corrupted by this contrast transfer function, and a lot of noise is added on top. Okay, and so our goal is to take this big pile of images, typically hundreds of thousands, millions of images, and to reconstruct the states on the left. And as I said, in principle, we need to reconstruct both the conformation and the poses for each image. However, I'm going to assume that I know the poses, so I'm going to make the problem simpler. This is sort of where the field is currently. In practice, this is done by running what we call a homogeneous or consensus reconstruction, where we ignore the heterogeneity and just try to align the images in the first step. And in the second step, we take the stack of images and the poses and input it in a pipeline like this, which is surely suboptimal, but this is where we are. Okay, the nice thing when we make this assumption is that there's no nice linear relationship between the x's and the y's. Okay. So I'll write down this linear relationship with a matrix PI, which models the projection and the CTF. Okay, even more ambitious is not to reconstruct a sequence of states, but reconstruct really a movie like this one that displays the range of motion that a protein can or biomolecule can take. So there's been a lot of different approaches that have been developed for this problem, but really all of them fit into a single framework, which works in 3 steps. So the first step is to embed the data set of images into a single coordinate system that we typically call latent space. Okay, so that's maybe a 2-dimensional or say 10 or 20-dimensional space. And the idea is that 2 images from their data set should be mapped to similar point in latent space, so close point in latent space, if and only if they contain a similar conformation. Okay, so assuming that we've somehow achieved this task, we can move on to a second step then, which is typically to take this latent space Z and to reconstruct volumes from it that are known by x's, and the volumes, we can visualize them, we can analyze them, and so on. And finally, there's a third step, which is to try to infer the motion in the data, and typically that's done by traversing the latent space in some way. So as an example, Cryo-Dragon that I mentioned earlier, which was developed by Ellen Zhang, who's also at Princeton in the computer science department. She's really the first to have introduced this variational autoencoder architecture for this problem. So in this case, there is a sort of 2 neural networks, one that takes the image and that outputs latent vector Z, and then another neural network, a decoder network that takes Z and outputs the volume. Okay, and that's trained end to end. So I'm going to talk mostly about the pipeline that Amit and I developed, which is not based on deep learning, but is based on statistical methods and sort of modern applied math methods. And I'm going to go through all 3 steps, but I'm really going to focus mostly on the latter 2 for time constraints. So just very quickly, step 1, which is this embedding step, taking this full data set of images and trying to represent it into some low-dimensional coordinate system. We do it using PCA, or principal component analysis. Okay, and the idea behind this is that we're somehow going to find a low-dimensional basis for the conformations, the xi's, given only images, the linear measurements. Okay, and I'm going to tell you in a minute some ideas how to do this, but let's suppose we've solved this problem and we found these principal components of the volumes, not of the images. Then what we do is we go through each image and can you see? No. So we go through each image and we try to represent the state inside the image as a linear combination of the mean and the principal components. Okay, and what we call the embedding, the Z vector is simply the coefficients of the expansion. Yeah, so that's how we take, this image, we find its 2 coefficients and we map it to this point. So that's what we call the Z embedding. Okay, so really the difficult part is finding the principal components in the first place. So a basis for the volumes. And that's difficult because we're trying to find the principal components of the volumes without actually observing the volumes. We only observe images, which are projections of the volumes. And so we have developed some algorithm to compute these principal components. Very quickly, the way it works is that we first estimate the covariance of the volume, which is something we can actually estimate directly from the images. And then once we've done this, we can extract the principal components as the eigenvector of the covariance matrix. And a lot of the work that we've done over the last couple of years has really been about trying to make this scale to the large data sets and make it robust to the high level of noise. Okay, so that was in maybe 1 minute how we embed the data set. And I'm going to mostly focus on the last 2 steps now. And the next step is to take sort of this latent representation and to reconstruct volumes. And to motivate the approach, I'm going to look at reconstruction of, let's say, a 1-dimensional heterogeneity. So let's do the simplest case first. So if we're trying to reconstruct a 1-dimensional shown our heterogeneity, we're really trying to recover a function of 4 variables. There's 3 spatial variables for the volume, and then there's 1 extra variable for the conformation. Okay, and if I fix the 3 spatial variables, and I look at a single voxel, then really I'm just trying to reconstruct a curve like this, right? So on the x-axis I have conformation, and on the y-axis I have, how the scattering potential changes as I move conformations. Of course, I don't observe that curve directly. All I observe is noisy samples. And what are these noisy samples? They're images. So really, each image will give me 1 noisy sample. And to be exact, on the x-axis, for each image, I will put their latent label. So I don't know where this is. Yeah. On the x-axis, we have the label that we computed from the previous step. And on the y-axis, it's really just the pixel value. Okay, and so even if I did a perfect job on the first step and I know exactly where to put them on the x-axis, there's still going to be a lot of noise on the y-axis just because the images themselves are very noisy. Okay, so now we have this noisy samples, and the question is, if I give you a particular point, a particular conformation here, how do you try to reconstruct its scattering potential there? And maybe the simplest idea one could come up with is to say, well, I'm going to look at a window around that point and just average the values of the points in that window. Okay? That's a description of kernel regression. And if you've decided to use kernel regression, there's only really one thing that you need to decide, which is like, how big am I going to make the window? So should I make a tiny window or a gigantic window like this? Okay, and to reason about how to pick the H, I'm going to turn to some of the known analysis of kernel regression. And basically it says 2 things. It says that if you want to minimize the error of your kernel regression estimate, you should take a big H when the SNR is low, okay? Or when the noise is very high. Okay, as an example, here in the blue curve, I took H, which is too small, and so I didn't have enough points, and it looks very noisy. On the other hand, I need to take a small H when the function varies quickly. So the example here is the green curve where I took a really large window, and because I'm taking a really large window, I ended up averaging all of the signal away, and I can get a nice reconstruction of the curve. Okay, so that's the general principle of how you should pick in kernel, you should pick your kernel width in kernel regression. So if you want to make this work as well as possible in Cryo-EM, we need to be cognizant of this because the SNR is actually highly variable in the spatial domain. So what that means is that if I'm trying to recover a low frequency, that's actually very high SNR. So I should take a small h, whereas if I'm trying to construct a high frequency, that tends to be very low SNR. And so that means I might have to take a big h. And not only this, but also in the spatial domain, how much the function varies depends a lot on where you're standing. So what I mean by this is that if I'm trying to reconstruct, this synthetic data set here, the tail is not moving at all. So in some sense, that means that I can just take the full data set to reconstruct this, whereas the head is moving a lot. And so when I'm trying to reconstruct the head, I have to take a smaller h. Okay. So the way we reconstruct volumes is just taking this idea and sort of pushing it to its limits, where we generate a sequence of kernel regression estimates, and then we tune the kernel width parameter very, finely over the spatial domain and the frequency domain. And we do this in a way that's completely automatic by cross-validation. So there's no user input here. Okay, so that's one way to generate volumes. It's maybe not the world's simplest algorithm, but it's also not very complicated in the sense that really all I'm doing in the end is taking a bunch of images and averaging their value. So the question is, okay, how well could this perform when you compare it to, a deep neural network or something like this? And for this, we are lucky because so Ellen, who made the Cryo-Dragon method, has developed this big benchmark data set where her and her group compared 10 different methods across 7 different data sets. And so what I'm showing you here is for each method and each data set, there's the reconstruction and the embedding. And here the color represents the true label. Okay, so if the color and the shape agree, that means that it's a good embedding. And So yeah, I'm going to show you all of the 7 data sets and their results. I will emphasize that I was not involved in this effort, so you cannot blame me for being biased. But they were nice enough to include our method. And in their paper, they have this nice little table at the end, which summarizes the quality of all the reconstruction across all the different methods and all the different data sets. Okay, and what they find is that the best performing method is ours in almost every case, across almost every metric, which is, I mean, quite remarkable because once again, our method is just doing PCA followed by some clever averaging. And a lot of the methods here are deep learning methods. They're deep learning methods that might use very intrinsic, complicated structural pairs or physics pair or biological pair. And yet very consistently, just PCA plus averaging will outperform all of them, which is pretty remarkable. So here's what it looks like on a real data set. So this is a ribosome data set where what I'm showing you in the corner is 2 embeddings, one recovered by a deep learning method called Dragon and one by ours. And actually in this data set, both methods do very well. They can recover the embedding quite well. We sort of have a good idea of what the labels on the images are for this data set, so we can test that. And here's a different state from the same data set. So both of them look quite nice, but if you're used to staring at Cryo-EM volume, you'll notice that the one on the right is actually slightly higher resolution. So if you sort of zoom in here, you can maybe spot the backbone here a bit better. And that's also confirmed by comparing it to the known atomic model for this ribosome. Okay, so I want to move on to the third step, which is how do we think about estimating motion from the data set. And really that's related to this great hope that we have in Cryo-EM that we can somehow reconstruct what people call a conformational landscape, okay? And a conformational landscape is not only reconstructing a bunch of volumes, but really showing a map like this that would point to where the metastable states of the systems are and how we can transition from one metastable state to the other. And in principle, let's say, we should be able to sort of compute all of this because it's really all dictated by the free energy of the system. And the free energy is a function of the density, as in the probability density of observing a particular conformation. So modulo a lot of sort of physics problem that, including sample preparation and so on, what you should be able to do is sort of just go into your data, count how many times a particular conformation happens, and that would sort of tell you what the free energy of the particular state is. Okay, so that's the hope. But if you run one of these methods on a real data set, and this is more like what you get. So you just get big blobs. You don't get like a nice structured map like this showing sort of metastable states. And so the question is, how do we reconcile these two pictures? How can we go from this to that? And to try to explain it, I'm going to show you some results on this synthetic data set. Okay, it's a synthetic data set that I made so that, okay, I know what the metastable states are, and it's also in realistic conditions like SNR and so on. And okay, so here I'm showing you the embedding that have been produced by 3 different methods. So recovery hours and then Cryo-Dragon and 3D are 2 deep learning methods on 2 different data sets. So the top one is the data set I just showed you with some synthetic heterogeneity. And the bottom one is another synthetic data set, but I scrapped out all of the heterogeneity. So in this data set, there is only a single state. But there are heterogeneity methods, so they'll try to predict some heterogeneity in the data. And what's maybe worrying is that at least qualitatively, if you look at, if you try to compare this, you'll see that it's not even clear that we'd be able to tell if there's any heterogeneity at all in the data by looking at the embedding, because both of them just look like big blobs. And in fact, it's maybe even more worrying if you look at the volumes that they produce. So these are, for the 3 methods, the volumes they produce on this data set that has no heterogeneity. So there is a single conformation, but they're heterogeneity methods, so they'll try to estimate some conformations. And what you see is that for Recover and Cryo-Dragon, these two methods, they will basically give you some weird artifacts, which is not great, but maybe not that bad. But then if you use 3D Flex, which is a neural network, which it tries to model explicitly deformation, it comes up with a completely physically believable deformation that looks wonderful, but is entirely hallucinated. Okay. So really, the issue here is that the images are very noisy, and as a result, the embedding is very noisy. And there's not much we can do about this short of somehow finding a much better microscope. But what we can do is try to at least model the effect of the noise on the embedding. And so that's what I'm going to do now. What we're trying to reconstruct is this conformational density, but we do not observe it, right? So this conformational density is what is dictating the free energy and so on. And that's what we're trying to reconstruct. But all we have is images. And really, in each image, there is a single sample of this density. Okay, but we don't observe the sample Z1. What we observe is actually just a prediction based on the image. And because the image is noisy, our prediction is also noisy, and so it's inexact. Okay, we repeat this over all of our images. And what we end up with is sort of a population of labels to which we can fit some kind of density. Okay. And now what is clear is that the density that we observe, the so-called embedding density, is not the same as the conformational density at the top. But the nice thing about the very simple embedding that we use in Recover is that because it's linear, we can basically track the effect of the noise. So we know what the noise in the image looks like, and so we can predict what the noise in the embedding looks like as well. And using this, we can then try to go back and take this density and deconvolve the effect of the noise on the embedding. Okay, by basically a normal deconvolution. Okay, this is what it looks like on this synthetic data set that I was showing you. So at the top, I'm showing you the 4-dimensional density, which is plotted over a pair of axes. It's very blurry because this is the density of the labels. It looks like very blobby, but once we've deconvolved it, okay, now we can see peaks of the density, which we would interpret as the metastable states, and then transition between them. Okay, and if we have successfully estimated this density, then we can go on to the next step and say, okay, now I know the density. Give me a trajectory from the star to the square that is low free energy, or let's say high density. Okay, and so in the software, we have a method to do this. It just tries to find this trajectory by solving some PDE, which is not actually quite the right PDE to solve, but it's one we can solve easily at least. Okay, so if I put everything together on this simulated data set, despite the initial embedding being very blobby, we can still recover sort of the density quite well. And from the density, we can recover the motion reasonably well, although not to extremely high resolution. Okay, so let me show you some examples on real data sets. So this is a spliceosome data set where after deconvolution, we can spot one peak of the density, which we interpret as the metastable state. And there's also a sort of very clear U-looking path. And so if we sort of take one point on each end and we try to find a trajectory between them, we end up with this trajectory, which looks like a motion of the car. Okay, here's a maybe slightly more interesting one, where this is another spliceosome data set, where if you look at the raw embedding, so here I'm showing the Cryo-Dragon embedding, but our embedding looks the same. It just looks like one big blob. But once we've tried to factor out the effect of the noise, we can actually find 3 peaks of the density, which we interpret as 3 metastable states of the system, which really corresponds to a different motion of the head and the arm of the complex. Okay, and then we can try and generate trajectories between these states by finding sort of high density path. And this is what our method comes up with. So you see that this motion between 2 states is maybe not the one you would have come up with, because it's sort of 2-step motion where first the arm of the complex is going down and then the head is going up. So that's what our method is telling. I'm not making any big claims about the free energy of this path, but this is what the method is. Okay, I'll give you one more example. So this one was performed by our collaborator, Wei Jianteng, and his postdoc, Jordan Muckle, and US Chicago. And so they're interested in this ACE complex for its role in cardiovascular disease. And so Wei Jian and Jordan, they were able to use Recover and actually use many different methods, like other heterogeneity methods and also molecular dynamics to try to understand this complex. And using Recover, they're able to find the metastable states and also the motion between them. Okay, so here are my takeaways. So in the low SNR settings of Cryo-EM, apparently, just averaging will outperform deep learning methods. And the Cryo-EM embeddings are very noisy, and we should be very careful about this because it can produce very believable hallucinations and can make inferring free energy or metastable states very challenging. And you can find my paper and all of my code on GitHub, and thank you for your attention. I'm happy to take any questions.
Very nice thought. I have a question about because it seems like there are two major components in the recover. First is PCA embedding, and then you do the code regression in the latent space. Seems like the two parts actually uncouple, it can be decoupled. Can you apply the regression to any other latent embeddings done by other methods, and you can see the improvement also?
Yes, so I actually have done this in the paper. I applied to a Cryo-Dragon embedding, and it actually does better than both Recover and Cryo-Dragon. Yeah, so you can do it. I think that in principle, it should work a little bit better on PCA embeddings because of distances are preserved, and really kernel regression uses distances. But apparently it worked. At least in this one case, it worked very well with the Cryo-Dragon.
I'm just curious on sort of compositional versus conformational heterogeneity, and just in general on these sort of continuous versus discrete state methods, on how people are sort of thinking about disentangling this, and if the sort of general view is that we sort of need a compositional filter first, and then we can sort of get a conformational that might be more continuous, or how you're sort of thinking about that.
Yes, so I think that, so first, most methods for continuous can be applied to discrete. Certainly mine can. In principle, it feels like it should be better to treat the discrete as discrete. So basically, the discrete versions use some kind of clustering instead of some kind of dimensionality reduction. And that seems to me to be the correct thing to do. Although it's not always so simple because you might have big classes and small classes, and the type of clustering method tend to be really biased towards big classes, and so people over the small classes altogether. Yes, and we need the so in some sense, it feels like if you had a very diverse data set, for example, multiple completely unrelated structures, and it seems that it would be better to do what you say, which is to first do maybe discrete and then do continuous if you want. And mostly because really the first step of all of this is to try to align all the images to a single coordinate system. And it becomes much more difficult to say what is the correct alignment if you have just a bunch of unrelated things.
So I have 4 more minutes for questions.
Yeah, I guess I'm curious where's the limitation on these methods? Or I guess, have you quantified or if you have comments on the performance of these sorts of methods as you go from lower quality to higher quality data sets, or data sets with lower versus higher number of particle images?
So I think that if you have a very low, how do you put it, quality data set, like you probably will not even sort of get to the stage where you can apply this method. So there's certainly like an absolute limit to it because, you know, the input from almost all of them is to have aligned images. And you can only align images given like sufficiently many images and sufficiently low noise. And then, so as you increase the size of the data set, so I mean, you would feel that as you increase the size of the data set, you know, of course, there's more computation, but the results would get better. But in fact, they don't keep getting better. So that's a feature of these, which is a little unfortunate, which is that most methods, including mine, are what's called, they're not consistent. So as n goes to infinity, the images goes to infinity, they don't keep improving. So there's a sort of limit there to current methods at least.
One more question, yes.
I just have a question about the correlation between experimental data and the method of preparation. Of course, a lot of Cryo-EM data are collected at low temperatures and a lot of warmer temperatures. Do you see any history? And of course, you're working on the more at the computational side, but in your experience, do you see a large effect of path dependence in terms of cooling methods or how the sample is prepared on the conformational ensembles that you see?
It's a good question, and I'm sure there is one, but I have no idea about this because I don't. I've never been in the lab or anything like this.
Yeah,
I know that people are thinking about this. I know much more about it, especially at top element.
Quick question. Now we can go to /blue. Thank you, Mark. So until 25 minutes. Yeah. Yeah, we'll resume at 10:15 from the coffee break, and we will give you details about the workshop photo in 1 minute.